So this is Kate Scow, and this is Soil Microbiology, Soil Science 111, and this is going to be the video podcast of Lecture 2, and I'm going to divide it into two parts. This is part 1. The lecture is about the soil environment or habitat and interactions of organisms in soil with their physical environment. What we're going to cover in this lecture is first give an introduction to soil as a habitat for life and soil as a habitat and how it influences microorganisms and other organisms in soil, but in turn how they create that habitat. Then talk about the distribution of organisms at different scales, at the pore scale, an aggregate scale, and at the column scale, and a little bit at larger regional scales. Then we're going to go into what are the different factors that regulate microbial activity, and hopefully have a class exercise that will help you interact with this information a little bit more directly. And then we're going to talk a little bit about ways that we look at the relationships of soil biology and their environment, talking about sterilization of soil as a tool that helps in this, and then talk about some other aspects as well that are some sort of second order, more complex issues with soil, as you would expect from soil and from microorganisms. Put them together and you've got complexity. Okay, so starting with this very simple diagram of soil, you walk on it every day. I don't know how many of you think about the fact that not only is it a solid phase, you don't necessarily sink in, but it also has pores in it. It has spaces, voids that are created between the different particles, and the sizes of these pores are a function of the sizes of these different particles and how they're put together. So soil consists of a solid phase, which is about half of the volume of soil will be solid, and that solid will include minerals and organic matter. That organic matter could be a very small fraction. It usually is not a very large portion of the solid phase of soil unless you're talking about very organic soils. But let's say 3% as an example here, 47% mineral. And then the other 50% of the volume of the soil is occupied by pores, voids. And these pores are filled with either air or with water. And that interplay between how much is filled by air versus water, which happens as rainfall events occur or you irrigate, very much changes conditions for microorganisms and for plants as well. And a very good ratio of air to water is about 50-50, so 25% of the whole volume of soil occupied by air and the other 25% occupied by water. This means that there's plenty of oxygen to support processes in soil, and there's enough water to also support those processes without waterlogging the soil. So that mineral fraction, let's look at that a little bit. The solid mineral fraction making up soil. We'll talk later about soil organic matter, not in this part of the lecture. So soil minerals, the soil solid is made up of 3 particle sizes: sand, silt, and clay. And this picture here gives an idea of the differences in scale between sand, shown over here, with a diameter of 2 millimeters going down to about 0.05 millimeters is the diameter of these particles. And then you can see silt. The next smallest one is in this range of 0.05 to 0.002 millimeter. And then clay, which is shown as just a speck here, orders of magnitude smaller than the sand, is less than 0.002 millimeters. That is less than 2 microns, right? And how big is a bacterium? Oh, there's nobody answering me. So yes, a bacterium is about a micron in diameter. So the clay particle is sort of at the scale of a bacterium, and there's also smaller clay particles as well. So what's interesting is not only this proportion of different particle sizes in soil, which by the way is called soil texture. Texture is the relative distribution of these three particle sizes in soil, and it has nothing to do with soil organic matter. Texture only has to do with the mineral fraction. But what's interesting is if you go over here and look at like the number of particles that you'd find in a gram of soil, and more importantly, the surface area. So you can see that the surface area of something like sand is really quite low, meaning that there's a very low surface to volume ratio, and therefore not much room for interesting things to happen, because in soil, most microorganisms are attached to surfaces, and all the good stuff in the soil, all the nutrients, and water as well, when things get really dry in soil, are going to be associated with particles. So around the sand, there just isn't that much surface area, but as you move into silt, and particularly into clay, you see this surface area just like mushrooming. Looking at over 10 to the 6 square centimeters per gram of like clay material. So why is that good? Sure, the surface area is part of it. There's a lot more places where microbes and chemicals and water can come together and do beautiful things together. But also clay is the product of a lot of soil forming processes, a lot of breaking down of larger particles that are over time broken into clay, and therefore there's a lot of modification as well chemically to the surface of the material of clay. So it's quite different chemically than a very inert sand particle. And because of this, clay has, for example, cation exchange capacity. We'll look at that in a minute. There's many sites to hold, for example, cations, interesting substances that microbes will use to and require for their life, and also organic compounds will be held there. So a lot can happen at the surfaces of clay, and some people feel life evolved from clays. Clays are pretty magical. Of course, none of these things are existing in their pure form. You're going to see interactions and reorganizing at higher levels of organization of these particles into things called aggregates, which we'll look at a little bit later. Loam, by the way, is a soil that has an optimal mixture of sand, silt, and clay particles, because of course pure clay itself is not going to do it either, because you're going to have a soil that will not drain water very easily, or things will not move through a clay soil very rapidly. So the best is this combination of sand, silt, and clay particles. So talking a little bit more about those surfaces, and particularly about clay surfaces, that ability to hold positively charged ions, and this includes a lot of important nutrients, calcium, magnesium, ammonium on surfaces, is the cation exchange capacity, and that's by interactions with the net negative charge that is associated with a clay surface. And this very much affects the soil physical properties and also the fertility of that soil. Organic matter as well contributes some of that cation exchange capacity. So that's also going to contribute as well to this ability that we're talking about here as well. So, I mean, to conclude, soils with a very low cation exchange capacity often are very low in fertility. So would you expect sandy soil to have high cation exchange capacity? That's right, you would not. There's not much surface area there, and there's not much capacity to hang on to nutrients. And in addition, sand has not gone through this weathering process and this chemical transformation that creates these this net negative charge and ability to hold on to cations. Okay, I want to throw this in. Microbial cells are charged too, but they are usually negatively charged at typical soil pHs. But they are attached to surfaces as well, not through cation exchange capacity, but through other types of chemical reactions like van der Waals forces, hydrogen bonding, maybe sharing electrons. But probably most important, like we talked in class today about extracellular polymers, this glue that they release, that's very important in sticking microorganisms to soil. And I think, as you may remember in class today, I said that it's very hard to separate, remove microorganisms from the soil. It's very, very difficult. You at most get 15-20%. You can do really horrible things with adding solvents and things like that, which probably end up killing them for the most part, and you might get a little bit more out, but it's really hard to separate microbes from soil. Okay, let's talk about the gas fraction now. We've been talking about the solid fraction. So, as you know, if the porosity, the pores that are created by this arrangement of these particles that we were just talking about, these pores of different sizes, if they're not filled with water, they're going to have gases in them. And in general, in a well-aerated soil, oxygen is going to be similar to the atmospheric concentration, 18 to 20% up there at the surface. And if there's a lot of good transport of atmosphere into the soil and out of the soil. As you get deeper in the soil, that oxygen concentration is going to decrease. But let me really make clear that it's not going down to very, very low oxygen concentrations, and it won't necessarily go into anaerobic conditions. It'll just go to lower oxygen concentrations. But it doesn't mean that everybody switches over to anaerobes there. What really is going to determine how low the oxygen concentration ever gets is whether or not there's carbon around, and therefore a need for that oxygen. And this you may find around roots, decaying organic matter, that dead vole that I always seem to talk about, I seem to be obsessed with. If there's a lot of carbon around, then a lot of activity is going to start up, and the oxygen in that area is going to be sucked down to very low concentrations. And at that point, it may switch over into anaerobic conditions. And what determines how rapidly that oxygen is going to be replenished that's used, let's say at 1 foot deep in the soil where that vole has died and there's all these microbes that are just working on that vole and using up all the oxygen, how rapidly the oxygen will get back and replace what they consumed at 1 foot deep, the oxygen coming from the atmosphere, is going to depend on the texture. So a high clay soil, it's going to be slow to get back, so then that anaerobic site is going to develop around that vole. If you had a really sandy soil, it may not actually ever even develop. Also, soil that's been compacted, that's had heavy machinery going over it, that's crushed the larger pores down into smaller sizes, that also is going to create a situation where it's not as easy for that oxygen to be replaced that's used. Okay, and this term redox, which you're going to meet in the flooded soil lecture and in this very cool lab that we do in this class on flooded soils, redox is a measure of the availability of electrons in a system. And that redox can range from minus 0.03 to plus 0.06 volts. We will revisit this later, but I just wanted to place this idea here in this context of oxygen. Other gases in the soil are going to include CO2, and that's usually going to be higher in the soil pores than in the atmosphere because of all this biological activity in soil. So you might see more on the order of 1% compared to 0.03% in the atmosphere, because microbes and plant roots are utilizing carbon in the soil and generating CO2. Again, how fast that CO2 leaves is going to be a function of the soil texture. There'll be other gases as well, most of them generated by microbial activity, like using alternative electron donors or acceptors. Sometimes these reactions, remember we talked about this in the first lecture, are going to create as byproducts these gases. And these gases include methane, N2 gas, nitrous oxide, N2O, I don't have that written here, hydrogen sulfide, H2S, rotten egg smell, right? These are all gases that you may find higher in soil than in the atmosphere, particularly where there's a lot of biological activity. Okay, third part, the water fraction. So where the water is, there will be less of that atmospheric phase, the gas phase that we just talked about. So how much water is present in soil is going to very much influence the exchange of gases. So we talked about texture, like a clay soil being slower, a soil in which it's slower for oxygen to replace itself. Also, if there's a lot of water in the soil, here's a picture over here showing some soil particles. This gray color is the water in an unsaturated soil. That means a soil that is not having all its pores filled with water. You'll see that there's big gaps where the air can pass through rapidly and you could have rapid gas exchange. When the soil becomes saturated, all the pores filled with water, you're going to have much slower rates of gas exchange. And then also an opportunity for anaerobic conditions to kick in. Because again, that oxygen from the atmosphere cannot come back and replace any oxygen that's used biologically. So water also controls the movement and concentration of nutrients. You can see the path length that like how far a nutrient that's, say, right here, my arrow, how far it takes, or how long it takes for it to get over to here on the other side. It would have to travel along this water film over those soil particles and over here, and that would take a long time. That could be a very long path length. But if you look down in the saturated soil, it's just a jump over this gap, just boom. And so that would not be a very long path length, and that nutrient would move much faster. So the diffusion path lengths for nutrients, like if microbes are over using a nutrient in one place, and for that nutrient to move, like extra nutrients then to move over via diffusion to replace what the microbe took up, it will be much longer path lengths in unsaturated soils than in saturated soils. Similarly, if microbes are going to move around, you can see it's pretty easy going for them to just swim across these pores. If they're filled with water, no big deal. They can turn on their flagella and just go across. But if there's not so much water in the soil, then they are stuck in these water films. Maybe they can still move, but it's a long journey from here, for example, over there. And I think I have a picture here of fungi. Remember, the fungi are really advantageous in that they can be growing here and they can send a hyphae across an air-filled gap over to another part of the soil to get nutrients there or water there, whatever they're after. Okay, and then also having more water is going to buffer temperature, so you're not going to have the big swings in temperature that you would in an unsaturated soil. Okay, soil water. So soil water is a pretty complex topic. I hope that I can simplify it enough that for those of you who don't enjoy the topic, you can kind of get the hang of it. But it's really quite interesting. And it's because soil water is measured best by the energy state of the water, something that we call water potential. And that's a much more useful and predictive measurement of soil water than just, say, water content, which would be like what you measure in lab, the gravimetric moisture content, how much water how much water is there, and you use an oven to dry it down and look at the difference in the weight before and after. Water potential measures the ability of soil water to move and or produce heat. So water potential is completely relevant to whether or not water is going to infiltrate and redistribute within the soil, or whether it can be removed from soil by evaporation or by plant uptake or by microorganisms taking up the water. And so what we call the total water potential is a composite of several component potentials. One such potential is the matrix potential, which is due to the absorption or the holding of water at soil surfaces or in soil pores. Pressure, which is due to an applied pressure, which you might have in a situation where the soil is waterlogged and you have like some water standing on the surface of the soil. Gravitational potential, which is associated with elevation and due to gravity's forces applied to the water in the soil, moving water downward. And then osmotic potential, which is the effect of solutes that are dissolved in that water, which sometimes get high enough so that then the potential changes as a function of that. So there's a very nice tutorial here at this website that I've got listed in red below if you want to go and get a little more background on this and also on the slides that follow. And I encourage you to do so if this is all a very foreign concept for you. Okay, so water potential is the potential energy, here's the definition, of water per unit volume relative to pure water under reference conditions. And we measure it in bars or pascals, like megapascals. So pure free water has a water potential of 0 bars or in megapascals. But most water in soil has a water potential that's less than 0, so it's going to have a negative number. So you can see that free water is water that doesn't have anything hindering it any way whatsoever. And in soil, water is usually not so fortunate to be free. Water has a lot of other things that are determining how much work that water can do, meaning whether that water can move or whether that water can be taken up by plants. And so this water potential is a measurement of that. Okay, let's keep going with this. So this is a kind of a ugly slide, but it has a lot of really important information here. So let's just think about soil that has experienced a whole lot of rainfall or irrigation, and this soil column here, which is shown in this upper part of the figure, and down below we have like a beaker collecting the water. So this is water that's recently been rained on, and what you see is that all the pores are filled with water, and gravity, and it's saturated, but gravity is pulling on that excess water in the soil, and so that gravity is going to help move the water out of that particular layer of soil and cause the soil to drain, so it won't stay as a flooded soil unless there's some compacted area underneath that's preventing that drainage due to gravity. So that's 0 bar would be this state of the water would be the water potential when it's at this state where all the pores are filled with water and it hasn't yet been subjected to gravity. So field capacity is this now condition of soil that's shown here, in which gravity has done its job on the soil and has moved all the water that it can move out, but against the potential of the soil to hang on to the water due to matrix potential, what we talked about previously. So field capacity is the water content remaining in soil. This is just a sort of a rule of thumb definition that remains in soil after it's been saturated and then allowed to drain for 24 hours. But also we can attach a pressure to this, an actual water potential measurement. And that's in the area of minus 0.1 to minus 0.3 bars is the number associated with field capacity. And field capacity is an ideal moisture content because you have some pores filled with air, some filled with water, and you have a lot of water that's being held as well. Then the next condition, these are sort of like cardinal moisture content, saturation, field capacity, is wilting point. So wilting point is the condition in soil where water remaining now is no longer available to plants. Like plants have like tapped into the water in field capacity and used it up. Maybe there's some evaporation, and this wilting point is the point at which plants can no longer extract the water present in soil. Microbes, by the way, still can access this water, but this permanent wilting point is from the perspective of plants. And so that is, the measurement at that point is minus 15 bar. Minus 15 bar is the wilting point. And this picture here shows you the sort of the conditions of the solid, which is going to stay the same, but the pore space. It's saturated soil, you can see it's all filled with water. Then field capacity, it's about, remember like that first slide, about 50-50 water air. Wilting capacity, there's still a little bit of water left, and then you can even get worse than wilting capacity, wilting point, which is the hygroscopic water, which is where water is, any water that remains is held very, very strongly to soil particles by adsorptive forces, and it can't be extracted by pretty much anything. And even a very dry oven, a very hot oven may not be able to remove all that water. You need a lot of energy to get that water out. Far more energy than a plant can put into getting that water out, or even a microbe. Okay, so, and available water is the water held between field capacity and wilting point. This water between these two water potentials, about minus 0.3 bar and minus 15 bar, is water, is available, soil water available to plants. Okay, how do we measure this soil water potential? One way is a pressure plate extractor, and this is a measurement of pressure or suction. And you may have seen this machine in Soil Science 100. And what you do is you take put small soil cores on top of this porous membrane that then you put inside the extractor, and then you apply different pressures to it. So you can, and you measure the moisture or the mass of the soil before you put it in the pressure plate, and it's always saturated when you put it in, and then you apply different pressures or suctions to that soil sample. So you might do it at minus 0.3 bar, remember what that is? That's field capacity. Or minus 15 bar, remember what that is? Wilting point. And after you do that, after you apply that pressure, you open the thing back up, take that soil column out. It will have had some water pulled out, and you weigh it after it's had that pressure applied to it. And then you can determine how much water is held in the soil at these different water potentials. So this is a very important way to get at those numbers of how much water content is associated with these different water potentials. All right, and then we get this kind of figure here, which shows you pressure in bars on this y-axis and percent soil moisture. And this is like using the oven to measure how much water is left. So what we have here then is at 0 bar, right? 0 bar on the y-axis. At 0 bar, we have as much water as is possible in that soil. That's the maximum capacity of the soil to hold water. So it's about 45%. And then as you apply greater and greater pressure, here we're going to about 0.1. Remember we said between 0.1 and 0.3 bar is field capacity. You can see at this point now we're down to about 32% moisture. And then here's more pressure's been applied, wilting coefficient, and you can see now the moisture is really low in the soil at about 8%. Okay, so you get this characteristic curve. You see this shape here. It tells you a lot about the soil. And then you can get even drier, put more suction on it, and there'll be less and less water. So this is a very important, useful curve that tells about how water is behaving in your soil. And how this curve looks is going to be a function of that texture that we talked about at the very beginning of the lecture, and also the organic matter content. And by the way, here are some of those terms that we talked about before, but this time mapped on this soil moisture release curve. Okay, so here's this relationship with soil water potential. Now this time we had the pressure here and the soil moisture here. This time we have the soil moisture content here, water content on the y-axis, and the water pressure. And sorry, the units changed, but this time in megapascals. But by the way, here's the conversion of bars to megapascals, and you can see that they only differ by 1 order of magnitude. That means minus 15 bars. The wilting point, minus 15 bars, is minus 1.5 megapascals. Okay, and by the way, there's our old friend field capacity at about minus 0.1 bar, or 0.3 bar. In here it's minus 0.03 megapascal. Okay, so you've got your bearings, hopefully. So let's look at this interesting relationship between these soil moisture release curves and texture. So here we have a sandy soil, right? And we start out at 0, no pressure, free water. So free water may, in a sandy soil, we may have something like, I don't know, 20% moisture. It's like 0.2, 20% moisture. And then as you apply pressure to the soil, look what it does with its water. It just gives it up. It just loses it. And so you don't apply much pressure, you get down to what's field capacity, and you've already lost a whole lot of water in the soil. And you can see here it's gone down to about, 8% or something like that. And what's interesting is you keep applying more and more pressure, and it doesn't have any more water to give up. It's already lost a lot of its water due to the at the lower pressure that's applied to it. Silt loam's kind of in between, and then let's look at the clay loam. Sorry, I don't have the whole picture showing here, but the clay soil, you can see, started out with a lot more water at 0, at 0 water potential, free water, and then as pressure is applied to the soil, it gives up some of its water, but it's not like a whole lot of water. And by the time, and so we're in the ballpark of like 38% moisture or something. Look at field capacity for a clay soil is almost like 40% moisture. Field capacity has tons of water in it compared to that sand that only has about 8% moisture in it at field capacity, at this best condition for plants to get water. And then as there's more and more pressure applied to it, getting down to wilting point, look at, there's still so much water left in that soil. It's like only a little less than 30%. tons of water in that soil, but that water is not to be had. Certainly not to be had by plants. And microbes will get some of that water, but not a lot of it, because those clay particles and these tiny pores that they create by the juxtapositions of their particles are holding onto that water extremely tightly, and it's very hard to get that water out. So if there was like a very hot day, and you had a lot of evaporation, you could see the sand is just going to give up all its water right away, but that clay is going to hang on to its water, and may survive and hang on to that water for a long time, but that water isn't necessarily available for biology. That water is not available to do work, as we say. And that water is not available really to move either, because it's going to be held really, really tightly to surfaces. Okay, I want to just talk about one other thing here. This column over here on the right-hand side shows you at these different water potentials. Remember, bars, here's minus 0.1, and then wilting point's going to be kind of in the minus 15 bars. Look at this last column. It shows you the maximum diameter. That means the biggest pore that will still be filled with water. The biggest pore size that will still have water. So at this field capacity, the biggest pore size that will still hold water is going to be on the order of 30 microns. This is just an estimate. It's a kind of a fantasy in a way about the soil, but it gives you kind of an idea that you're going to have a lot of pores at field capacity that are big enough for microbes to live in. Okay? But when you get down to wilting point, down into the minus 15, we're having the largest size of pores that can still have water in them are going to be smaller, and can get so small that they're going to be too small for microbes to live in. So you can see, like in this clay soil, there may be a lot of water around there, but microbes may not be able to have access to that water. So that's the point I want to get across here, is that how that water is arrayed, where that water is found at this small pore scale is very important to how much life is going to be able to take advantage of that water. Okay, so another concept. That's the end of soil water for now. And please let me know if you need help on thinking about soil water. We will, I guess what I didn't talk about was the sponge metaphor. And maybe I can find you a video on that to use that as a way to also think about soil water. Okay, so another concept that's very important is soil structure. So soil structure is higher order organization of those particles that we've been talking about, and soil organic matter and pores. And this structure, as we'll talk about in a minute, is very much, and we talked about this in class, is very determined by soil biology, the architects that I keep harping on, that very much contribute to creation of structure. So we have the most extreme case down here, is like a sandy soil that does not have good structure at all. It just has a lot of individual grains that don't stick together and are, just, there's no organization there. It's just texture. And then we have this perfect, or this best situation over here, that when we go out to Russell Ranch, and I'll bring some aggregates sooner, where you have this granular soil structure that is very biologically derived, that you have this nice sort of, they say cookie crumb here, but these really nice aggregates that have a lot of organic matter in them, and they have a lot of room for air and water to move among them and inside them as well. And then you've got these other kinds of structures, blocky. Actually, you kind of see this in our conventional soils out at Russell Ranch, where they don't get much organic matter. Also prismatic, columnar, platy, these different kinds of structures that also can reflect the parent material for the soil as well. Okay, and I guess if we move up in scale, so we went to the aggregate scale, but also you're going to find at the landscape scale, you're going to find different kinds of soil profiles that are going to be a function of how much activity and weathering and time has been in how much time they've been exposed to, and also the soil forming factors that contribute to creating soil, how much they've had time to play. So my point here is just as you move across a landscape, you're going to run into many different kinds of soils with very different horizons at a bigger scale. And I'm not going to talk much more about this, but this can very much influence. You can see if you have a soil that looks like this versus this, you can imagine big differences in the biology of the soils. Okay, so I now want to talk a little bit about soil as a habitat and distribution of microorganisms or microbial communities in soil. Now that we've talked about sort of the ingredients of soil in the what I've lectured on so far. So we can think about the distribution of microbial communities at 3 scales. At the pore scale, which is going to be from the micron to the centimeter scale, the pore and the and we'll include aggregates in here. The soil column scale, which is going to be more at the centimeter to meter scale, and the landscape scale, which is meter to kilometer scale. And I think I've touched on this before as well, is that in microbiology, soil microbiology, we're often asking questions at really different scales and then trying to integrate across these different scales. So at the scale of an individual microorganism, or let's say a consortium, which is probably more likely, we're talking very small scale pore, and this is where the microbe is interacting with its electron donor, its electron acceptor, its carbon source, and it's metabolizing material, it's producing new products, it's demanding, it's consuming oxygen in that area. The oxygen needs to move back to them. But this is at the scale where things are actually happening. And then we have, it's interesting to think about microbes in soil as you move down with depth into the soil profile. We have a very different kind of habitat at the surface of the soil than we do deeper in the soil, and we'll talk a little bit about that. And microbes play a big role as well in creating these differences that we see as we move into soil in depth. And then we have landscape scale phenomena that, particularly when you're talking about things like respiration and production of methane or consumption of methane or nitrous oxide production, we're often interested in these processes at very large scales, particularly with climate change, and trying to make predictions and trying to mitigate climate change through manipulating some of these processes. We are very interested in these larger scale phenomena that are created by microorganisms at these smaller scales. So we ask very different questions at these scales, and I think that's my main point there. So if we look at soil, this is someone I think went a little bit crazy with imagining these different kinds of microenvironments within the soil, but I still think it's interesting to think about this, that we have depending on where plants are growing and where organisms like some of the soil fauna have shaped the soil, have started to create this, like heterogeneity and this kind of architecture in the soil, you have some very different microenvironments that can be quite different in terms of the numbers of organisms present and the kinds of organisms that might actually be present. This is a blow up here. This thing that we're calling a drillosphere is a microenvironment that's created by a worm that's moving up and down, pulling organic matter from the surface down deeper in the soil. And it will create, it'll bring some of the organic matter down deeper in the soil. We talked a little bit about bioturbation, the ability of tillage to occur, but do the organisms in the soil, and they're also, these earthworms are also going to create feces and casts, which are going to also redistribute the organic matter that in this profile of this soil, creating this really different kind of environment where then you may have these different processes. Here's some gases being produced at different locations in this drillosphere. Over here we have the porosphere, which is the name that's given to the soil around the plant root. We see some aggregates and big pores and small pores inside aggregates. Someone has named the aggregate the aggregatosphere, where then you're going down inside this aggregate and looking at the clay and this organic matter and bacteria perhaps hiding out in these small pores, and fungi maybe going by and gluing together some of these particles, helping make this aggregate. So another really different kind of microenvironment. And then we have the rhizosphere around the root here that's blown up here, which is this very special environment that we'll devote a lecture to, that has a lot of carbon available around it, and again, a very different kind of habitat for microorganisms than you'll find in what we call the bulk soil, which is in the area that isn't under the influence of the plant root. Okay, and there's this very famous quote by Martinez Bierjink, that is a highly debated quote, but it's a really interesting one that we continue to debate even as our ability to answer the question improves. And it's everything is everywhere, and the milieu or the environment selects. And he was saying this with respect to microorganisms, that pretty much microorganisms are widespread throughout the environment. You can find most kinds of organisms in most places, but who's actually going to be showing up and engaged and being active are the ones that are going to be selected upon by whatever the environment is offering at that moment. So it could be, if it's suddenly very wet and anaerobic, it might select that group of organisms. If there's a lot of carbon that goes in, there might be a lot of decomposers coming in. If there's elevated temperatures for some reasons, it may select another group of organisms that weren't showing much evidence of their existence beforehand. And this is this idea of everything is everywhere and the environment selects. It's not necessarily true, and of course it also depends at what scale of phylogenetically you're talking about, because sure, there may be a fungus everywhere and a bacteria everywhere, and the environment selects which one, but I mean that's at a very coarse scale of resolution. At the species level, maybe there are a lot of species found throughout the world, a lot of the same species, and indeed this is what happens when you start to look at soil. But they may end up being actually really different subspecies that are adapted and all present in these different kinds of environments that you see in the world. But anyway, the reason that this is of significance is that it has a lot to do with whether or not inoculation is an important thing to do. A lot of people debate that it isn't the absence of organisms that is preventing a certain kind of process from occurring. It's because there's something in the environment that isn't allowing whoever is there, the native talent, to express themselves. And what you need to do is focus less on putting that organism in, because the organism might be there, the organisms that you want to do the job might be there, but what you need to do is think about what's limiting them. And this will come up again in the course, but it's analogous to, like, with humans, human health, probiotics versus prebiotics. Like, should you change your diet to, like, improve your gut, or should you add organisms to your gut? And I think it'll be interesting to keep talking about this in the course. It's definitely a big and important area to think about. Okay, I wanted to give you this picture to to show the relationship between microorganisms and these different particles and pores that we were talking about. So we were going from very small, micron here, here's nanometers, very, very small on the right-hand side to bigger, getting up to like 10 millimeters. And here's two kinds of soils: a sandy soil that has a large portion of its pores being very large pores, and not many small pores, in contrast to a loam that's going to have a nice mix of very small, medium, and large pores. Defining pore sizes as the fine pores are below a micron, medium pores are in this 0.1 up to 10 micron diameter, and large pores 10 and above microns. Okay, so we have this contrast between these two types of soils. Then we can think about the organisms here. So we don't have organisms until we get to viruses. Viruses are very small, as you remember, and they could be associated with these fine pores. And then as we get into bacteria, they're going to be occupying these medium pore sizes. And so there's going to be more opportunities for them in a loamy soil than in a sandy soil in this medium pore size. And then protozoa and fungi getting bigger. There's going to be not so much room for them in the soils that have more of their pores smaller, but there's going to be more room for them in the larger, coarser soil, we call it, textured soil, like a sandy soil. But on the other hand, those soils are not going to hold water very well or very long. So there's going to be that play between there being bigger pore sizes, but they may not hold the water. Anyway, just trying to get you to start to think about this relationship between the texture that we've talked about before, the pore size, the water, and the distribution of different size organisms. And what isn't shown there are the bigger organisms that, remember, they're in the first lecture in that table at the end of the lecture, and you can see that things there, organisms there, are getting quite large, up in the order of millimeters and centimeters, and you can start to think about how they're going to be held in the soil. Of course, if they get really big, they can just start to drill their way through soil as well. And of course, they are architects as well. They'll change the soil properties as well. Okay, aggregates. Aggregates, I want to talk now about how microbes play a role in making soil aggregates. So it's an important structural unit of soil. It's made of particles and pores. And microorganisms, remember we talked about extracellular polymers or polysaccharides today in class. And this is a cross-section of a colony in soil. It's a French one and it's inverted. But you can see here some individual bacterial cells. There's maybe the mother, and she divided into her daughters. And you can see there's only a total of about, 10 or 12 different cells in this colony, in contrast to what you see on a Petri plate, where you have these massive colonies of billions and billions of cells. In soil, colonies are much, much smaller. And you can see here that this mother and her daughters are enmeshed in this extracellular polymer, in this material that's been excreted by the bacteria, which I always say most self-respecting soil organisms are going to be sitting in these polymers. And remember, we talked about in the lab how people try to select the particular, like from a culture, if you have a liquid culture in the lab shaking and you see this clumping together of cells stuck together by these polymers, you're going to try to push those aside and go for the clear stuff, go for the organisms that aren't making extracellular polymers, and therefore getting rid of a really important trait in nature, and perhaps losing some of the information about that organism because you're selecting for a strain that doesn't make this polymer. So anyway, the other interesting thing you can see is this array of mineral particles around the colony, so that there's almost like this entombing of this colony sitting in this extracellular polymer by clay particles that are attracted to the surface of it. And this kind of interaction between the clay and these extracellular polymers, and eventually these dead bacteria that are also going to decompose and go into making soil organic matter. This is how soil organic matter is made, is by these interactions, because soil organic matter is really soil organo-mineral matter. I think this is just a beautiful picture that lets you really think about how this is actually happening at the scale of the microbes. Here's a big beaker full of goop. It's a fungal extracellular polymer, so it just looks totally like glue as well. And when you look at your rhizobia on your plates in the lab later on, later in the quarter, you'll also see that rhizobia produce a lot of extracellular polymers, maybe helping them associate with plant roots or hang around near plant roots when they're not actually in a symbiotic form. Maybe helps them stick around. Okay, so these aggregates, we'll look at some in class very soon. Here's one that is a conventional aggregate. It hasn't gotten a lot of carbon, and you can see a lot of the angular structure of the minerals making it up. And this organic one is like a big fuzz ball. There's so much carbon that's gone in that system, a lot of aggregates that have created like a coating of organic matter around it. But aggregates are really interesting because once they get bigger than a certain size, they're going to have oxygen diffusion limitations. It'll be slow for oxygen to move inside the aggregates, and so that you can have this heterogeneity in the soil created by creating these really different kinds of microenvironments that in this case inside are going to be anoxic or anaerobic. Microbes in there are going to have to use, other than oxygen as electron acceptors, for example, use nitrate or use sulfate and produce these other kinds of gases and be involved in biogeochemical cycling in different ways. And the larger the aggregates are, the more anaerobic these sites are. And we'll talk more about this as we go further in the class. And this is a very famous picture by Tisdall and Oates that shows, again, scale. Here we have very fine clay particle size, and we on the order of 0.2 microns, and then we go up to about 2 millimeters to a macro aggregate size. And what you can see is from this diagram, it shows also, here's the different sizes of organisms along this scale. And then here are some of the different processes that are important in creating these soil aggregates, which I've already told you has a lot to do with the extracellular polymers. So, at a very small scale, you may have chemical interactions holding together clays, but then you've got microbial debris and extracellular polymers that are going to interact with the inorganic compounds at the order of 2 microns and start to create these little nuclei. And then you're going to have hyphae coming in and holding together some of these smaller bundles together into larger aggregates. And plant roots are going to play a role as well, particularly as you move larger into the micro aggregate size and then the macro aggregates size. So there's this continuum of sizes and aggregates, and there's a lot of, as you can see, biological processes that are playing a role in binding together these aggregates. Sorry, I think aggregates are incredibly important and interesting, and I do tend to go on about them, but hopefully you'll be able to look at them in class pretty soon. Oh, there's another aggregate picture. Okay, just showing you the same thing, but a prettier picture. It's showing you exactly what I just showed you before. Sorry, there's a little bit of redundancy here, but it's worth thinking about it. This is one of the most important principles in soil that have to do with the interaction of microorganisms and soil physical properties. Okay, and then here's just some pictures thinking about, it's really hard to draw soil and to think about how organisms array themselves within it, but this picture to the right sort of shows you some roots going around an aggregate, and then these little pores inside, bigger pores where there might be a mite, and somebody who's going and looking around and trying to then prey upon these bacteria that are held in these smaller pores that are, shaking with fear, but safe for the moment as that mite goes by, and they're going to be held in smaller pores. And in fact, soil that has smaller pores is able to provide a refuge for more organisms to survive against predation than in a soil that has only big pores. And so we have also fungi moving around. They're going to not be in the really small pores, but they're going to be in the larger pores, particularly where they're putting these fruiting bodies and things like that. Okay, going up in scale, we have the soil column scale. So I want to talk a little bit about the distribution of organisms in the soil column scale. This is a generic soil column, and in general, we always have these different horizons in the soil, but of course it's depending on the particular kind of soil, how actually well developed these horizons are and whether or not they're all there or not. But here's a generic typical soil that has an O horizon at the surface, which is the organic horizon, then the A horizon, which is a mineral horizon, but it's going to be very much influenced by the organic matter, which of course is coming from the surface, from the plants that are growing at the surface and decaying and introducing organic matter into the soil. But this A horizon is more mineral, but very influenced by the organic horizon. And then we have the B horizon, which is deeper in soil, and it's less influenced by the organic material at the surface. It's going to be higher in clay content, higher in aluminum and iron, and but an area where there has been a lot of transformation of the original parent material. At the C horizon is where you're going to find much more the manifestation of what is the original geologic material in which that soil is formed. There, that's going to be highly mineral, not much organic matter at all, high bulk density as well. And then below that, you're going to hit the regolith, the R, which is the consolidated parent material that the soil was created from. Okay, so as you move deeper in soil, you have decreases in organic matter, decreases in oxygen, but remember, it's not going to go down to zero. It's going to depend on how much carbon is there to consume it, how low the oxygen will go, but it will be lower than the horizon. You also have decrease in light. That's kind of like a duh. So any photoautotrophs are going to be really declining as you move deeper in the soil. And then higher clay content and higher bulk density as you go deeper in the soil. So what about the distribution of microorganisms as you go deeper in the soil? Well, it's pretty straightforward, and we can look at these different pictures. Here are some different groups of organisms, and this shows you the relative biomass concentration, very high on this side. Everyone starts out high at the surface, but as you move deeper in the soil profile, you get numbers of organisms or biomass of these organisms dropping off. The algae drop right off, the protozoa drop off, the bacteria and fungi are going to go deeper in the soil. I don't know if the predators quite drop off that much, but you can see in general you're going to have most of your microbial biomass at the surface of the soil, and by the time you get down to, I would say, 10-15 centimeter rule of thumb, does depend on the soil, but you're going to already experience a big drop in the biomass as you go deeper. Here's another graph that shows you numbers of fungi and bacteria at these different depths. Here's the counts, and here's the centimeters of deep of the soil. Sorry that it got cut off, but it's centimeters. So you can see a huge drop in numbers of bacteria as you move from 0 to 10 to 10 to 35 centimeters deep. And this is pretty consistent. Your biomass really drops as you go not that deeply into soil. And here's activity as well. Activity measured as some pesticide degrading. And we have the pesticide degrading over time, 100%. And then this is how much remains over time after it was applied. This is the fastest, right? It shot right down and degraded really quickly. That was in the surface layer of the soil. But you can see in the deeper soil, it takes much longer for that pesticide to degrade, or any kind of activity is going to be lower because your numbers are lower. Think about that with respect to biodegradation of contaminants in soil. Like where your talent is, where your capability is going to be in that surface layer. And there's been tons of barrels of terribly toxic chemicals or petroleum that are deeper in the soil. They're like sunk into the soil profile, often in areas where there's very little biological activity, and they don't biodegrade, whereas they might biodegrade in the surface. Okay, here's a term that's really important to know, the vadose zone. So the vadose zone is this area of soil, but we're distinguishing it from soil that is very much under the influences of plants and all the biological activity that we're talking about. But the vadose zone is still unsaturated. So the vadose zone sits on above the groundwater table, right? So there's a lot in the news about groundwater and aquifers. So in the groundwater, deeper in the soil profile, right, it means that all the pores are going to be filled with water because you've hit the water table and it's groundwater. So that's saturated. But you have this zone of unsaturated area between this soil, this surface layer that is this biologically active, high organic matter soil, and the water table, which is called the vadose zone. And you may also have this capillary fringe, this area that the water table moves up and down in depending on the season, that is a little bit more nutrient rich and maybe have a higher moisture content. So as you go into the vadose zone, you see lower numbers. Remember, we already looked at that in the first picture. but in the previous picture, but I do want to say is even though the numbers drop off, there's still organisms present, and those organisms stay present all the way down throughout the Vadose, through the groundwater. 4 miles deep, they've found organisms like deep, deep in deep subsurface zones, but definitely much lower numbers in biomass and activity than in the surface. You tend to find more organisms associated with clay and silt, not a big surprise, and more at these interfaces, at the capillary fringe zone or where the texture may change in the soil. Okay, landscape scale. So there's lots of interest in what we call biogeography of microorganisms. Is it true everything is everywhere and the environment selects? And particularly this has been really like energized by the availability of new tools to be able to look at microbes in nature. So definitely if you use plate counts, you will find most groups of organisms in most soils. You'll find indeed that everything is everywhere. But with molecular tools, you can look at this a little bit more rigorously. And as I said before, the answer depends on the taxonomic resolution. Like what scale, what taxonomic scale are you looking at it? But some of the things that we're finding out are that, you know, microbes do vary across landscapes, and people are really trying to understand, is it because of the plants that are growing there? Is it because of the soil texture? Is it because of the parent material of the soil? Is it because of the climate? Is it because of something in the chemistry? And there's no clear answer. I mean, this is like looking at, are there tundra and alpine and grassland type microbial communities? You know, like we see plants, like we're able to find these plant communities in these different regions, and we have some understanding of what determines whether you have this kind of plant community versus that. People are asking those questions about microbes. And so some people think, oh, it just depends. They follow the plants. It depends on the plant species. But that's not true at all. They're independent of that. Like the kinds of communities that form are independent. They interact with the plant species, but plants don't determine it. What seems to be some of the things that are important is pH. pH seems to be a major driver in terms of determining whether a community looks one way or another. Also, climate is important, and land use is really important. For example, if you agriculturally manage a soil that was before a grassland, that's going to have a big impact on the communities, and whether it's a forest or under a driveway, under a road, with plant type and latitude not being as important. And I think it's also really interesting to think about, like, so why, but why isn't it super clear cut? Why don't you have these really distinctly different communities, like growing in different kinds of elevations or in different latitudes? And I think one of the key issues that we don't have a great understanding about is dispersal. There's a great potential for microbial communities, soil microbial communities, to be dispersing all the time. And one vehicle for that is dust, is soil particles suspended and being blown away to a different area from where they came from, and microbes riding on this dust. And there's been some really interesting research showing soil particles, but also microbes coming in, like from the Sahel Desert in Africa, finding organisms, and definitely the soil in Florida, and also Mongolian soils coming to Lake Tahoe. There's times when like a huge amount of the deposition of dust that you might find it, over a short period of time, they can relate it back. They can prove that it comes from Mongolia, from some big, climatic event that lifts up what is probably really eroded soil due to overgrazing And obviously coming along with the soil are a lot of microbes. So there's a lot of like continual mixing of microbes and introduction of microbes from other areas to new areas that you kind of have to deal with as you try to think about this issue of biogeography of microorganisms. Okay, we're going to stop there, and I'll pick up the rest of the lecture in the next part.